Hello everybody, welcome back for another exciting lecture. Today we're going to start tackling chapter 5, which is entitled Cellular Movement and Muscle. Now, the whole purpose of this chapter is going to be to take a look at the different proteins and components that cells can utilize, in essence, to control their overall cell shape, move items within their cytoplasmic content, but also as a whole can be used for things like motility, as well as locomotion. And towards the end of the chapter, I should say towards the middle, we're going to take a look at it on the grand scale, how we can utilize these components to explain muscle contraction and relaxation. Now, the quote-unquote machinery or the items that we'll use for cellular movement will tend to all evolve around cytoskeleton and the motor protein. The cytoskeleton is mostly composed of proteins that often form the intracellular network that you would find inside of the cell. This is where you would look at things like your microfilaments, your microtubules, and your intermediate filaments. Your motor proteins, these are going to be a group of enzymes that we can use energy from ATP to move items around. So we'll talk, for instance, about one of the motor proteins named myosin, which is essential for forming the thick filaments in muscles, which allow us for the sliding filaments to be discussed so that we can see our muscle contraction and partly our muscle relaxation. Now, there are three ways to use the skeleton for movement, and that's going to be either almost like a little roadblock, and I love the illustrations that are on the side because we can see right here that we can use our internal components of cytoskeleton that we will find inside the cytoplasmic of the cell, that we can either use it almost as a mapping / road component so that we can move around our different hormones, our different secretions. We can do our exocytosis. We can also utilize it in essence to reorganize or organize the skeletal network as a whole. So this would be towards the overall shape of the cell. The fact that the plasma membrane has fluidity to it, that the cell is not a rigid structure, that you can have items that are kind of like ammoid movement where you're extending within the environment by making these pseudopods. And then last but not least, we can have a very organized type of locomotion where we can use our skeleton almost like a little rope that we can tug at. And as we're tugging at it, what we see happening is that we can go ahead and have items like muscles contract. And when we stop tugging, those muscles will go ahead and relax. This will play a role in our sliding filament theory that we'll be tackling throughout the mid part of the chapter. Now, the first item that we want to look at within our cytoskeleton is going to be microtubules. For microtubules, and you know what, just so everybody's sure, let's go ahead and take a look at the picture that's at the bottom of our PowerPoint. Microtubules are going to be these extensions, these tube-like polymers that you can find all over the cytoplasmic content of the cell. And the way it's going to work is that it's mostly composed of this protein called tubulin, hence the name, microtubules. They come in different isoforms depending on what cells we're looking at and what their overall function will be, but all of them will be located within the cytoplasmic content. And what we see happening is almost like the spokes on a bike wheel. They will go ahead and radiate out from the center and connect with the the plasma membrane. So on your illustration, what you're going to notice is very lightly in purple, I'm going to go ahead and circle it, you're going to see the nucleus of the cell. And then you have a darker structure in orange. That structure is going to be called your microtubule organizational center. And what we see happening here is that we're going to use that as an anchoring point for the microtubules to attach. And from there on, the microtubules are going to radiate outwards so that they can connect with the cell membrane or the plasma membrane. And what we're going to notice is that the polarity of the tubing will be different. The negative end will be towards the microtubule organizational center, the MTOC, which is near the nucleus or the center of the cell, whereas the positive end of the microtubule is going to be interacting with the plasma membrane. You want to pay attention to the polarity because in a little bit we're going to talk about how you have different motor proteins that you can utilize to move up or down the stream, and they're going to use the polarity as their flow of direction. Now, let me give you a little example of how we can use these microtubules to conduct our intracellular transport. So to move items that are actually inside the cell to kind of move them around. And as you can try to imagine, we can use this, for instance, to move around our different organelles to get it nicely and reorganized. But in the case that we're going to do this, we're going to take a look at the African clotto frog. And what we see happening with the frogs is that they are quickly able to change their skin color. They can go from a very light toned skin color to a much darker tone. How do we explain the fact that they can do this so quickly and that they have this phenotypical plasticity? Because the color change is often regulated by changes in their hormonal level. More specifically, this particular hormone called the melanophore stimulating hormone. And what we see happening is that the melanophore stimulating hormone will go ahead and produce these granules of pigments called melanophores inside the cells, and they can either distribute the pigment all over or they can cluster the pigment in batches. And the way the pigment is going to be moving is along the microtubules. What do I mean with this? Okay, go ahead and take a look at your picture and take a look at your top frog. And what we see happening here is that this frog tends to have a relatively light skin tone with darker patches distributed all over. Under the microscope, we can see that the nucleus or the center of the cell tends to be filled with lots of melanocytes, different granules of color. Now, how do we explain this? Well, if we take a look at our microtubule setup, what we see happening is here is our plasma membrane. Here is going to be the center of the cell, so closely to the nucleus by the MTOC. And we see happening here that all our melanopores, all our pigment granules are going to be clustered in the center, thereby allowing all the cytoplasm right over here that has no exposure to the granules to go ahead and show a light tone. And where the granules are clustered together, those will be the dark spots that we see happening on our first example of our frog. Now, how do we go ahead and switch from light to dark? Well, how about we take our melanopore cells with our granules and we take our microtubules and we allow the pigments to start distributing themselves all along the length of the microtubule. As they're doing that, they're spreading their color, their pigment, all through the cytoplasmic content. And what you end up seeing is instead of having clusters in the center of the cell, the entire cell gets a beautiful dark shade to it. So you can see how the pigments are now evenly distributed throughout the cytoplasmic content, from the MTOC in the middle all the way to the plasma membrane at the end. And as the pigments distribute, you can see it here from a histology point of view, the way the phenotype will alter is to go ahead and darken the skin tone of the frog. Isn't that cool? And the granules are able to obviously migrate back, be spread out, and they'll just keep using those microtubules almost like a little roadmap for distribution pattern. Now, how exactly does your body make these microtubules, and why do you have a positive and a negative end? Well, it turns out that your tubulin that you're looking at, the protein that makes up these microtubules, it's actually a combination or a dimer of alpha and beta tubulin. So what you can do is go ahead and take a look on your right-hand side of the screen, take a look at your illustration, and you're going to notice that you have your alpha and your beta. And what we're going to see happening is that they go ahead and form spontaneously. They're able to pair up and make their alpha and beta dimer. And as they're doing that, we often see that there's some phosphorylation going on the beta end that doesn't occur on the alpha end. And because of that, you then create a polarity, which usually indicates that the alpha will have the negative charge, where the beta will go ahead and indicate the positive charge. And it turns out that since this is occurring spontaneously, as long as you have access to the alpha and beta, they will continue to link up together and initially will form what we call a protofilament, which is more like a string. And if you get enough of them, they start bunching together in sheet format. And if the sheet is wide enough, it eventually will roll up in a tube, and that will then give way to our microtubules that we will lay out all across the cytoplasmic content of the cell. Now, the positive and the negative end seems to be correlated also to which end is more likely to shrink or grow. So what we end up seeing is that the negative end has a tendency to shrink, meaning that it loses some of the tubulin, whereas the positive end tends to be more of the growing side. And part of that is because it has an easier time attaching new dimers on that end. So if you look all the way at the bottom of your illustration, you notice that the shrinkage is going to be more towards the negative end and the growth more towards the positive end. And this is going to lead to asymmetrical growth, since one side is more likely to grow, where the other side is more likely to overall shrink, all due to the polarity differences between the alpha and the beta. Now, there are different items or factors that will come into play that will stimulate the overall growth and breakdown of the microtubules, but probably the most important factor will be the availability of tubulin. Tubulin will basically be a direct correlation to if you'll have growth or shrinkage. And what we see happening here is that we have a little graph that shows us how our concentration of tubulin can affect both the positive and the negative and showing you the CC values. Now, the CC values are your critical concentration. And that's basically the concentration of tubulin that's needed for you to have a zero growth rate, meaning that it's the amount of growth equals the amount of breakdown or release. So the length is nice and consistent. And what the graph is trying to show you is that the positive end of the tubulin is a lot more sensitive to tubulin. Let me go ahead and reword it, my apologies. What this graph is showing you is that the positive end of your microtubule, my apologies, is way more sensitive to your tubulin levels, because you can see right here it's at a lower concentration, and look how it shoots up. It quickly goes ahead and encounters its critical concentration volume, but any slight shift or changes to the tubulin level will go ahead and promote extensive growth. On the other hand, the negative end of your microtubule, it takes a lot longer for it to reach its zero mark. Before that, it's doing a lot of shrinkage. So it requires much higher levels of tubulin for the negative end to grow. And most of the time, the cell does not have access to that much tubulin. So that is what partly why we explained that at levels of tubulin, it is more likely for the positive end to grow due to its increased sensitivity for a growth pattern, whereas the negative end will only grow if you have very high levels of tubulin, and in most homeostatic situations, it will go ahead and have shrinkage that will occur on that end. On the right-hand side of the illustrations, you're going to see another representation of our cell with the nucleus right here. We see our MTOC segment, and it's showing you your microtubules radiating outwards. This is also the part where it's showing you that if you were to zoom in, you would notice that the microtubules, by the time it gets to the plasma membrane, will actually utilize MAPs, or microtubules associated proteins, right over here, to form almost like an anchoring point or a connection point so that they can be physically attached to the plasma membrane. We can also utilize the MAPs as connectors from one tubulin to the next as it's distributed. So it's a fantastic way to kind of stabilize the microtubule in place and allow it to have interaction between the plasma membrane and itself. Now, along the microtubule, we also see that we have some motor proteins that we can utilize as movement vesicles. So we can kind of fill it up with content, hormones, or any type of secretory production, and we can allow them to move up and down the microtubule. And a lot of this will be determined by the polarity as well as the type of motor protein that you're looking at. When it comes to microtubules, you have two motor proteins that will consistently pop up, and those are going to be your kinesin and your dynein. Now, your kinesin tend to move towards a positive direction, whereas your dynein will move towards a negative direction. Now, you can go ahead and reference in your node, but remember that when we look at our microtubules, the negative end tends to be towards the MTOC or the nucleus, and the positive end tends to be towards the cell membrane. So that means that kinesin will tend to move from the center of the cell towards the outside by the cell membrane, where dynein will move from the outside towards the inside. All of this will be fueled by the ATPase enzyme that they are able to hydrolyze within their structure. And depending on how much ATPase or how large the ATPase domain is, will control the overall movement and rate of movement of these motor proteins. Let's go ahead and take a look at this from an illustrative aspect. All right, so right over here we have our nucleus. We have our MTOC, and now remember the negative end is towards the center of the cell, and then we have our positive polarities making its way over to the cell membrane. All right, and now on the illustration right here, they say what we can do is we can utilize these motor proteins. The first one we can look at is kinesin. Kinesin, remember, tends to move towards the positive end. So that's the one that we, for instance, can fill the vesicles with items that we're trying to secrete out of the cell. Maybe certain enzymes, maybe certain hormones, anything you can really think about. And they will go ahead and walk down the microtubules, and in the case of kinesin, it will walk towards the cell membrane, so we can obviously maybe do exocytosis and release the items outside of the cell. The one at the bottom is going to be your dynein, and dynein usually will tend to carry empty vesicles because it's returning the vesicles back to the MTOC area so it can be refilled. Obviously, it could also have items that it brings back to the cell. But either way, what we want to remember about dynein is that it tends to move towards the negative end. So it will allow you to move from the cell membrane back over to the MTOC area. Please keep an eye on your polarity, and please keep an eye on the movement type of our motor proteins. So for our microtubules, we have our kinesin and our dynein. Kinesin moving towards the positive end, towards the cell membrane. Dynein moving towards the negative end, back over to the internal workings of the cell, over to the MTOC. Another way that we can utilize microtubules is for the construction of cilia and flagella. Cilia is usually fine hairs. They tend to be bundled together in large masses, and in the human body, we love to have cilia in any of our orifices. So for instance, if you take a look at your respiratory system with the oral cavity and the nasal cavity, if you take a look at your urinary system and our reproductive systems, all the openings for the body that are exposed to the internal environment, or to the external environment, excuse me, they will be filled with cilia because cilia is able to trap pathogens, or for instance, if they become wrapped in mucus, it's able to trap it and it will move in a wave-like formation. And we can utilize that movement to kind of eject the pathogen or any type of allergen out of the system, preventing it from entering the body any deeper. For instance, in our respiratory tract, we have this thing called the mucociliary escalator, and that is a collection of cilia that literally lines the throat region. And if you ever, for instance, inhale maybe an allergen, like maybe a dust particle or some pollen, or even a pathogen, we don't want that to get into the lungs because it might go ahead and cause things like pneumonia. So what we see happening is one of the first lines of defense is that pathogen will get trapped in mucus, and the cilia will move in an escalator fashion, tracking the mucus upwards with the pathogen until it gets to an area where you can either cough it out, sneeze it out, or you can swallow it, allowing it to be dropped in the esophagus, moving its way over to the stomach where the acidity will most likely destroy it. Now, in addition to the cilia, we can also utilize microtubules to set up the flagella. Flagellas are usually longer than cilia. You usually just either have them singled or pair. They are found in a wide variety of cell types. Bacteria love to use them as a mode for locomotion. Gentlemen, you are the proud owner of the only cell in the human body that has a flagella, and that is the sperm cell. Ladies, don't feel left out. We have the largest cell that the human makes. That's our oocyte. There you go. So be proud of that. Now, when you think about the flagella, I want you to think about a whip-like structure. And what we see happening is that it moves in an asymmetrical pattern. And that is then why the cell can utilize it perfectly for locomotion. Now, when you look at the anatomy of the cilia and the flagella, what we see happening is that the microtubule proteins will be arranged in what we call an axoneme. And an axoneme usually indicates that you have an external ring and you have an internal working. So go ahead and take a look at your illustration on the right-hand side, and illustrated in green are our microtubules. And what we see happening is that on the outer ring of our structure, we see that they tend to be bundled together in little doublets. Okay, so here's a little doublet, and if you count, you're going to notice 9 of them to make a full circle, so to cover the entire diameter of your flagella in this case, or the cilia, same concept. And on the inside, you're going to have singlets, so one by itself, but they're going to be, go ahead and they're going to have 2 of them. So this is where you come up with the 9+2 ratio. So this is the general setup or arrangement of the microtubules to give shape to the cilia and the flagella. And an interesting thing that point out is that the dynein motor protein will go ahead, and you can see the dynein right over here interacting with the microtubule. What the dynein motor protein will do is that it will activate the microtubules, or it will drag the microtubules in a side-to-side motion. And because it activates one side only compared to the other, we see that we create almost like this wave-like, whipping-like motion. That's part of why I have that animated sperm cell just whipping its tail. And so imagine that if you're looking at your insides of the flagella, we would only activate one side, causing the flagella to move in that direction, and then we would quickly turn that side off and activate the next side, causing the flagella to move in the opposite direction. So you keep going side to side, side to side, side to side. And this asymmetrical activation is then what gives rise to that stereotypical movement of a swimming sperm cell. Now, another category of items that we can utilize for overall intracellular transport or locomotion is going to be microfilaments. Microfilaments are composed of a different protein compared to microtubulins. Microfilaments are going to be composed of actin. Their motor protein of choice will be myosin. And oftentimes we'll see that microfilaments will be sort of like the hero or the main concept discussed when you talk about muscle contraction and relaxation, aka the sliding filament theory. So let's go ahead and dive into microfilaments now and see how they contribute to cellular movement, both inside the cell and when you look on the outside towards the entire locomotion of the organism. Alrighty, so as I mentioned to you before, the microfilaments are composed of actin, and what we see happening is very similar to our microtubules. This will be a spontaneous form of growth. However, what we see happening here is that the actin will initially start off as G-actin. Now, the G stands for globular, meaning that they'll be circular, and what we see happening is that they'll start polymerizing, and as they cluster together, we then go ahead and categorize them as F-actin. So we can see it right over here As it's elongating, you'll have your F-actin. Notice also how you'll have different polarity sites. You'll have a positive and a negative end, and very similar to what we saw in the microtubules, the positive end is more sensitive to growth than the negative end. There are also some key terms that I want to point out over here that can be abided to both microfilaments and microtubules. One of them is treadmilling. Now, when you walk on a treadmill, you're basically walking without going anywhere. So treadmilling indicates that you have growth and disassembly happening on either ends of the microfilament and in microtubules as well, but you're not changing the overall length of the filament. So whatever you're adding to the positive and whatever you're taking off from the negative, they will cancel each other out and you'll maintain equal length of the filament. So you see right here in my illustration, if I add 3 more G-actins, I also remove 3 actins from the negative end. So this is treadmilling. You see growth and assembly on both ends, on either ends, I should say, excuse me, but the overall length stays the same. If we see that the filament wants to increase its overall length, oftentimes what it will do is it will try to go ahead and increase stability on the negative end, and it can do so by adding a capping protein. When you add a capping protein, you basically make it harder for the negative end to disassemble. So it stays nice and stable. And then what's happening on the other end? Well, the positive end is more likely to grow. So we can utilize that for a growth pattern of the entire filament. Our microfilaments within the cell can be arranged in many different ways. In a little bit, I'll show you a picture of how we can do what we call a tangled network, or we can do more of a bundle setup. And as you can see from the PowerPoint, you'll utilize different proteins depending on the setup. The tangled network will tend to be linked by the filament protein, where the fascin protein will come into play when you do your bundles. And don't worry, I'll show you what I mean with this in just 1 second. We also see that you'll have a protein that will able to link the microfilament to the cell membrane, just like we saw it with our microtubule, and that will be the dystrophin protein in this case. And this one we often see in many different functions. So for instance, it's been studied excessively in the microvilli in the digestive tract. It will help with the overall cell shape, and it can be used for movement. And this part of the slide just kind of talks about the fact how the dystrophin protein is once again interacting with the intracellular component and linking it to the cell membrane of the particular cell we're looking at. So here is our eukaryotic cell again, and here's our nucleus. Now notice how unlike the microtubules, the microfilaments is a lot more numerous in its quantity. It is all over the cytoplasmic content, so we see all these lines that are distributed throughout. And if we start over on our right-hand side, it talks about the fact that in general what we can do is we can take our actins and we can bundle them together. So this is our first arrangement type that we do our bundles. And when we do the bundles, we're going to go ahead and use the fascin protein to link them together. On the other hand, you can have a network, a tangled network that forms. And when you do your tangling or your network formation, you're going to use your filament as a cross linker. So notice how the shape is slightly different, because in this case, we're not really making bundles, we're just crisscrossing to make a network. On the left-hand side, you're going to notice that it also highlights the fact that your microfilaments will make physical contact and will be anchored to the cell membrane. And instead of using the MAPs, which we saw in the microtubules, your microfilaments will go ahead and use the dystrophin protein as the cross-linker in this case. So there's a lot of similarities between the microtubules and the microfilaments, but there's also very small differences, and especially structural and support differences. So definitely make sure that you highlight these as we're kind of perusing through our PowerPoint, and I am pointing them out the best that I can. All right, let's take a look at that motor protein that's associated with microfilaments. Let's take a look at myosin. All right, so myosin is going to be extremely important because it's going to allow us to start talking about something that I keep mentioning, which is the sliding filament theory. And what we see happening with myosin, it does comes in lots of different isoforms. It all depends on what cells we're going to look at. So for instance, if you take a look at the illustrations on the right-hand side, you're going to see myosin 1 and myosin 5. These myosins tend to contribute more to like intracellular traffic, whereas myosin 2 right here, this particular isoform, is the one that we're going to use for muscle movement. So this is the one that's going to help us form our thick filament of our muscles. Now, regardless of which isoform you're looking at, you're going to notice that from a structural aspect, they have three distinctive regions. They have a head section that is going to be utilized for ATPase activity so that we can convert energy from the ATP molecule into mechanical energy so we can utilize that for our movement options. You're going to have the neck, which will allow for the movement of the head. And then you will also have your tail section. Your tail can be used for many different things, including binding to the subcellular components. Now, when we utilize our setup of our myosin, and remember the myosin 2, the isomer that we're going to use for our muscle myosin, what we're going to see happening is that part of what we need to do, if we want to utilize it for the sliding filament model or the contraction of muscles, is that we need to go ahead and make the actin and the myosin interact with each other. And it's almost going to be like a pull and grasp type of routine. So it sounds a little silly, but on the slide, I have my little mime right there, and he is motioning that he's grabbing a rope and he's tugging at it and he's releasing it. But in a little bit, when we talk about the sliding filament theory, I want you to keep that motion in mind. And what we're going to talk about is the fact that the microfilament, the one that's made of actin, will serve as your rope, where the myosin, your power protein, will represent your arms. And what we want to do is, if we want to have our muscles contract, we're going to go ahead and have to engage the head of the myosin with the actin. And the head of the myosin is what's going to make physical contact with the actin, and it will allow the actin to slide over in a very controlled pattern. Now, we can't let go of the actin when we're contracting, so what we're going to see happening is that most of the time, one head of the myosin will stay intact, will stay physically connected, while the other head will let go so it can easily tug over the actin. So that's what our little mime's trying to do. He's trying to hold onto the actin with one head of the myosin, pull it, and then take his other hand, which represents the second head of the myosin, grab another piece of the actin and pull it again. So you have this whole grasping, pull, and release mechanism. And as you're listening to me and you're going, what is she talking about? Don't worry, I have a lot more scientific illustrations coming up that will highlight the sliding filament theory. All right, how about this illustration right here? Okay, so we are looking at the interplay between the myosin and the actin. Okay, so the actin is going to be represented by these little dark lines, and then the myosin is going to be represented in orange. And the reason the orange is the myosin is because, as you recall, it has a head section with the neck and the tail. So those little orange beads that you see moving, those are the head of the myosin. And what I was trying to tell you on your previous slide is, when you look at your muscles and you contract your muscles and you relax your muscles, the way that we're going to explain that motion is through the sliding filament theory. And that basically means that if I were to zoom into your muscles, I were to see that on a cellular level, I need to get my actin and my myosin to do two things. I need them to do a chemical reaction, and I need them to do a structural change. The chemical reaction basically means a cross bridge. I need the myosin head to make physical contact with the actin binding site. So that's my first step. I need to make that physical contact. And there's my mime reaching with his hand to touch the rope. So the myosin needs to make physical contact with the actin. And that process is called a cross bridge. Once you have your cross bridge, now you need to get the filaments to slide over each other, because as they're sliding, what we see is that the muscle starts to contract. And that's what my little illustration right here is trying to show you. That we are making physical contact, so the orange to the black with the myosin heads, and that we are sliding them closer, and we're going back to our normal position. As you slide closer, that is your contraction, and as you go back to your original position and you extend, that is your relaxation. Now, the sliding, the actual movement, is going to be our structural change, and that is what we're going to call a power stroke. And a power stroke is going to entail that some of the heads of the myosins are going to have to let go of their cross bridge, cock their head, and reach out to another binding site so that they can pull the actin, they can pull the rope closer to their point of origin. And when they do that, they're sliding. People used to think that when you contract the muscle, that you actually shorten or compress the muscle, but you don't. When you contract the muscle, what you're doing is you're changing the overlap between the actin and the myosin. And the more you allow them to overlap, the stronger the contraction becomes. But in order for them to overlap, they will first need to make physical contact, which is your cross bridge, and then they need to start sliding. And the sliding part is going to be your power stroke. Now, there are lots of requirements needed to get all this done, but one of the most essential items that will be needed will be good old-fashioned ATP. ATP will be our chemical energy that we're converting into energy of motion. And what we see happening is that we need ATP actually both to contract and relax. And we notice the importance of ATP when we see that, unfortunately, a person becomes deceased. When a person becomes deceased, they stop producing ATP. And as a result, what we see happening is that they can develop rigor mortis. Rigor mortis is when you run out of ATP and the head of the myosin can no longer relax from the actin site. So everything kind of clenches in place. And what we see happening is that usually within 3 to 12 hours of death, a person will stiffen up. And that stiffening up is the fact that you're able to do your cross bridge, but when it was time to do your power stroke or even to relax, there was no more ATP, so it was locked into position. Rigor mortis is not permanent. Usually, if we give it another few hours, the actual filaments will start to fall apart and degrade by themselves due to the fact that the person is deceased. And when that happens, the entire filament structure falls apart, which means that there are no more cross bridges and definitely no more power stroking, and the fingers and all the other muscles will relax. So here is another illustration showing you our sliding filament theory. And I really like this picture because it will highlight the interplay between the actin and the myosin. So before we start reading all the different boxes, let's go ahead and make sure that we know the players that we're looking at. So over here in red, I have my F-actin, right? Here's my actin. You can see them coiled up, my positive and my negative end. And notice how the actin has these little black spots. Those are going to be your binding sites, okay? And then here in yellow, looking like a little golf club, that's my myosin. So I have my actin in red and I have my myosin in yellow. And what this one is trying to tell you is that if you want to get your muscle to contract, you want to initiate the sliding filament model, then the first thing we're going to have to do is we're going to go ahead and have to do our cross bridges. So we're going to make our physical contact between our actin and our myosin, which we can see right over here. And I want you to pay attention what they did. The first contact you made, notice how they went ahead and they colored that actin molecule a pink, just so you can kind of keep track of where that first initial contact was. Once you make your cross bridge, what we see happening is that one of the heads of the myosin is going to detach, and in order to do that, it's going to utilize an ATP molecule. And as it detaches itself, it's going to extend, and it's going to reach out to another binding site. So see how we went from pink to orange? It's just showing you the secondary binding site on the actin. And once it finds another binding site, it's going to go ahead and do its power stroke. And the power stroke basically means that it's going to cock the head back and it's going to allow the actin to slide. So before the pink was over here, now the pink is all the way over here. And as it's doing it, that's the same mechanism that you're seeing over here on my little illustration. The fact that it's making contact, cross bridge, and the fact that it's doing the power stroke. It's sliding over each other. And you're going to need to have ATP to keep this process running. And as long as you can keep sliding, you can keep contracting that muscle. Another few items that we can kind of take a look at is if we want to calculate exactly how much movement the myosin head does, we can calculate this thing called unitary displacement. And that basically calculates the distance that the myosin head will move from one cross bridge to the next. So from one physical contact to the next. This will all be dependent on the overall length of the myosin neck, as well as where the binding site of the actin is located. Because as you might recall from our previous structure, you might have noticed that the binding sites were not lined up in a linear fashion. They're more of like a little helical structure. So that's why I put the little picture of the spiral staircase, because the binding spots will align in a very helical pattern. And what the myosin now has to do is it has to bind to one, and when it power strokes, it has to try to reach for the next one that it can find. And that distance is then what we're going to call our unitary displacement. And like I said, the location of the binding site and the head of the myosin head, those are the two main factors that will play a role in exactly how far we can move. We can also calculate what we call the duty cycle. The duty cycle is when you're interested to figure out how much time is spent making a cross bridge versus how much time is being spent crossing from one bridge to the next, so engaging in your power stroke. And usually what we see happening is that a typical myosin head will have a duty cycle of around 50%. And that means that half of its time is spent with the cross bridge, the other half of it will be spent making its power stroke motion. And we know that this is important to point out because as we are doing our sliding filament theory, we want to remind ourselves that we have multiple myosin heads that are in play that are connecting to the active sites on the actin. And we can't have them all removed at the same time because that will loosen the connection and allow the muscles to forcefully relax. So you will always need to have a certain percentage of myosin heads that are actively engaged with the actin and obviously doing their power stroke so you can go ahead and have your muscle contraction. Take a look at this picture right here. So we have our actin right here in red, and you see how the actin sites are in helical patterns? They're wrapping around, and then here's our myosin head. And basically what we're doing if we're calculating the unitary displacement is that we want to see what is the distance that they move from one side to the next. And that is then overall calculated. And like I said, the two factors that will determine that will be the length of the neck as well as where exactly the next binding site will be on the actin. Let me go ahead and show you this slide and then we'll kind of take a break from this chapter so we can kind of process all this information before we go into part 2 of the chapter. This is basically another way of representing your actin and your myosin. And in the next part of the lecture, we'll take a look at the histology of muscle cells so we can kind of remind ourselves of the striations that we might have previously seen when we learned about our skeletal, cardiac, and smooth muscle. And within those striations, we see that we have a thick and a thin filament. Well, the thick and the thin filaments, those are nothing more than your myosin and your actin. We know that muscles are composed of muscle cells, which are your myocytes, and inside of those myocytes, you will have your contractile elements. And these contractile elements will be your thick and your thin filament. And you already know about all this because if you take a look at the picture, you're going to notice that your thick filament is nothing but myosin, clustered together, wrapped around so that the heads are exposed, looking to bind to the active sites on the actin. And the actin is going to be represented by the thin filament. So here we go. It's a polymer of the actin. And what we see happening is that in order to stabilize the actin, we will utilize different proteins, including CAP-C on the positive end, tropomodulin on the negative end. And we're also going to have to talk about the troponin-tropomyosin complex. This complex will allow us to control when the actin and the myosin make physical contact with each other, aka when the muscle is able to contract. So I'm going to go ahead and stop on this particular slide. Take your time, peruse through the material. As you know, I'm available through email and checking the discussion sessions. And whenever you feel like you're ready, let's move on to the second part of Chapter 5, where we will continue our discussion on muscle cells, and we'll take a further look at those thin and those thick filaments. All right, talk soon.